Many major scientific and pharmaceutical discoveries and advancements have come from nature. Phytocannabinoid compounds derived from the Cannabaceae plant, such as CBD, have attracted significant interest for the treatment of multiple diseases and disorders. Kannalife has spent a decade understanding the CBD molecule and its therapeutic potential. We have taken a data-driven scientific approach to the discovery and development of novel molecules like CBD. We have made significant improvements in the chemistry and efficacy of CBD-like molecules that are up to 200 times more potent, 10 times more bioavailable, 5 times safer, and 1,000 times more effective. In short, it's CBD 2.0. It is more drug-like and target-directed than CBD. Kannalife's peer-reviewed and published research on its leading target drug candidate, KLS 13019, has produced a clear understanding of its mechanism of action. We have focused on the treatment of chemotherapy-induced peripheral neuropathy and other oxidative stress-related disorders involving neurodegeneration. Kannalife has been issued U.S. patents on composition of matter and utility for its novel CBD-like molecules, including its leading target drug candidate, KLS 13019. KLS 13019 now stands ready for clinical evaluation to treat CIPN, a progressive and often irreversible condition affecting about 1/3 of all patients undergoing chemotherapy. Opioids are the current standard of care for patients suffering with CIPN. KLS 13019 offers a non-addictive therapeutic alternative for the treatment of CIPN, chronic pain management, and neurodegeneration. To better understand the toxic impact of chemotherapeutic agents, let's focus on the peripheral nervous system, the PNS, and the dorsal root ganglion, otherwise known as the DRG, where toxic chemotherapeutic agents accumulate and produce peripheral neuropathy. The sensory neurons in the DRG contain synaptic mitochondria. Inside these synaptic mitochondria is a complex called NCX1, a sodium-calcium exchanger that regulates intracellular calcium levels and acts as a calcium rheostat, upregulating or downregulating calcium in the neuron. This provides protection for the neurons by regulating calcium levels critical to neuronal health. Too much or too little can cause a toxic response from the cell, cause peripheral neuropathy, or ultimately result in the death of the neuron. Paclitaxel is a chemotherapeutic medication used to treat a number of types of cancer. Paclitaxel can be toxic to some sensory neurons by increasing oxidative stress and calcium overload. Let's find out how and why KLS 13019 works in the treatment of CIPN. The target is NCX1 that resides in the membrane of mitochondria. Paclitaxel leads to an increased toxic amount of calcium. KLS 13019 works by stimulating NCX1 to help the mitochondria remove the calcium overload to provide a therapeutic benefit. To understand how this works, we know that if NCX1 is decreased in concentration, KLS 13019 no longer protects sensory neurons from chemotherapeutic agents. This is a classic means of determining a drug target. Our research and development efforts in cannabinoid therapeutics, with KLS 13019 as a leading target drug candidate, also holds great promise for the safe and effective treatment of neurodegeneration, chronic pain management, and other oxidative stress-related disorders. At Kannalife, we're deploying the most creative and innovative scientific research minds from academia and biotechnology to discover and bring to market novel cannabinoid therapeutic agents and medicines for patients with serious unmet medical needs.